Charles Darwin argued that all humans belong to the same species and speculated that our origins would eventually be traced to Africa. In his book, The Descent of Man, Darwin wrote, "In each great region of the world, the living mammals are closely related to the extinct species of the same region. It is therefore probable that Africa was formerly inhabited by extinct apes closely allied to the gorilla and chimpanzee. And as these two species are now man's nearest allies, it is somewhat more probable that our early progenitors lived on the African continent than elsewhere." Today we know that our evolutionary past is traced to Africa. Not only did the earliest hominin bipeds emerge in Africa, along with the first human toolmakers, and later our ancestors who began to cook their food, but our genes also first emerged in Africa. Recall, our early modern lineage traces its ancestry to Homo heidelbergiensis. Not only did Homo heidelbergiensis lead to the evolution of modern humans, but they also diverged into our closest hominin ancestor, our cousins, the Neanderthals. With respect to Homo heidelbergiensis, paleoanthropologists have recovered fossil evidence of our ancestors that date back some 600,000 years until they became extinct some 100,000 years ago. They lived in Africa, Europe, and Asia. Important fossils include the Bodo skull in Ethiopia, the Florisbat skull in South Africa, and the Broken Hill skull in Zambia. Modern humans evolved from Homo heidelbergiensis in Africa. Evidence of our origins are traced to early modern African residents some 300,000 years ago. Some of the important modern Homo sapiens specimens include the Jebel Irhoud skull in Morocco, the Kibish and Herto skull in Ethiopia, and then a Globa skull in Tanzania. By analyzing human DNA, molecular anthropologists are also able to trace the genetic origins of humankind to Africa. But before we explore our genetic journey, we need to know a little more about haplogroups, haplotypes, and single nucleotide polymorphisms. Humans have over 3 billion base pairs in our DNA. If you could stitch all of the DNA of a single person together, it would stretch from the Earth to the Moon and back some 8,000 times. In examining DNA, anthropologists study haplotypes, or the genetic material that is inherited from a single parent, matrilineal haplotypes and patrilineal haplotypes. Over time, through the process of evolution, haplotype blocks emerge. These blocks are areas that tend not to mutate and are transmitted from one generation to the next with little to no recombination. Haplotypes are best understood as the genetic variations on a chromosome. For example, here we observe 4 unique blocks on a DNA strand. It is also important to understand that certain areas on a chromosome can be more or less likely to support mutations. Some areas on the strand of the DNA, for example, are hotspots for mutations. Single nucleotide polymorphisms, or SNPs, refer to a mutation in DNA that causes nucleotides to change position. For example, DNA is formed from 4 nucleotide bases: adenine, cytosine, guanine, and thymine. With a SNP, the nucleotide cytosine, for example, may trade positions with thymine in a particular area, and these SNPs are inherited by subsequent generations. Because SNPs change the structure of DNA from an original population at the genetic level, populations eventually become somewhat distinct. Remember, although a population carries the original haplotype blocks that they inherited from their ancestors, these new SNPs create variation. When new SNPs begin to accumulate within a population, new haplogroups emerge. In other words, molecular anthropologists, for example, can see the genetic variation between populations because of the presence or absence of a SNP. Again, as time passes, new SNPs emerge. For example, here we observe the original haplotype blocks that were present in the original populations, the first known SNP to emerge, the green highlighted area, and a new SNP highlighted in orange. Because SNPs are located on very small areas of DNA, and DNA is very long, remember there are some 3 billion base pairs, they only produce a small amount of genetic variation, even though our physical appearance may look different. This is one of the reasons why, although there is variation among human populations, we are between 99.7 and 99.9% genetically identical. In the end, the analysis of human genes allows anthropologists to trace the origins and spread of humankind. The earliest maternal haplogroup is referred to as L by molecular anthropologists. As the L haplogroup underwent genetic changes, subclades, or variant genetic groups emerged as humans migrated away from Central Africa. Eventually, as the earliest humans migrated eastward to Ethiopia and the Sudan, one of the new subclades produced enough genetic variation to produce a new haplogroup that we refer to as M. Importantly, the M haplogroup represents the genes of the first African women to migrate out of Africa and into Southwest Asia. Eventually, a new haplogroup emerged, N, and these people are those that began to settle in Asia. Haplogroup D refers to all those people who also carried the first genes of humans in Africa, the woman who migrated from Africa to India and the Middle East, then to other areas of Asia, and finally migrated to the Americas, carrying the genetic markers of all the populations that they're connected to. Keep in mind that there are too many haplogroups and subclades to discuss here, but they all illustrate one fundamental truth. All humans are genetically related, and our earliest ancestors lived in Africa.